Hello, my name is Trenton Owens. I'm a graduate student at North Carolina State University. I'm a member of the microbiology graduate group. I am a member of the Rhodanobacter lab. However, what I'll be speaking about today is my work back at Lawrence Berkeley National Laboratory as a member of the Deutschbauer lab, where we develop functional genomics tools in bacteria. We were part of the Enigma project funded by the Department of Energy. Now, the Department of Energy is very interested in the Oak Ridge Field Research Challenge. And what this site is, it is a legacy nuclear waste site where nuclear waste containing nitrate was pumped into these aquifers, these wells. And that nitrate is incredibly corrosive. It lowers the pH significantly, and that causes leaching from the surrounding environment, leaching of very toxic heavy metals like cesium, chromium, arsenic. It's basically got the entire periodic table dumped underneath these wells, and it's a terrible place to live. However, one organism seems to thrive, and that would be Rhodanobacter. So one particular species, Rhodanobacter denitrificans, was isolated from these wells. It's unique in that it is capable of full denitrification under highly acidic conditions. And that's very interesting because if you're trying to remediate these very contaminated aquifers, you would want organisms that could play a role in lowering the soluble concentration of this terrible pollutant. And we have seen that organisms like Rhodanobacter that can lower the nitrate concentration can play a role in also lowering the solubility of uranium. And there are researchers who are interested in, for example, adding carbon sources to these wells to spur the growth of these heterotrophic bacteria and lower the soluble concentration of uranium so that it stops leaching to surrounding aquifers. However, if you want to do that successfully, you need to understand the metabolism of these organisms. And for that reason, you need genetic resources in Rhodanobacter, which is what I aim to do. However, it wasn't easy. Rhodanobacter is highly resistant to transformation. What we do is we develop transposon mutant libraries, and that starts with making individual transposon mutants, which we were incapable of doing, at least not at scale. So in light of Rhodanobacter's native restriction system, we hypothesized that our transposon delivery vectors were being degraded upon transformation. So restriction enzymes were eating up our delivery vector very much like Pac-Man trying to finish the level before that delivery vector was capable of expressing the transposase and landing a transposon in the host genome. However, after PacBio sequencing of the genome of Rhodanobacter, we noticed that there were methylated motifs within the genome that were also present in our unmethylated vector. And so by redesigning the vector to avoid these motifs, by simply changing 9 nucleotides, we were able to see almost a 100-fold increase in transformation efficiency. And that allowed us to successfully create an entire representative transposon mutant library with mapped transposon insertions all across the genome. Now, what we do is we take those mutants and we pool them together to make the library, and we allow those mutants to compete under the same sort of stresses that were present within those contaminated aquifers, with the idea that if you're missing a gene that was relevant to growth under stress, then we could track that loss of fitness through the barcodes themselves. And so what we're looking at here is actual fitness results from our BAR-seq assays. And this is just one example. This was looking at Rhodanobacter from the perspective of one of its genes, something that's predicted to be an arsenic resistance gene. And so what we observe is that out of the 216 different fitness assays that we did with this Rhodanobacter library, 208 of those fitness assays resulted in ultimately no effect whatsoever when it comes to this particular gene. That is to say, loss of function in this gene had no effect one way or another. However, in the 8 experiments that we did where an arsenic-containing compound was present, these mutants would do much worse than their peers. So with a fitness score between -4 and -5. Now, what's next is to be able to explore all of the stressors within this contaminated aquifer and each individual gene's contribution to surviving under that stress, including combinations of stress. And so this technology is a powerful way of determining what genomic features enable success in different environments. Thank you very much.